last session, I will show some recent research experiment that they has introduced the silage into the animal so they can uh, serve animal digestibility as well as animal production. So before going to the detail, I would like to uh, provide some questions so you can enter some like A, B, C, or D in the chat box. So my question, like overview. So first, start from the what is the primary goal of the silage process that we need? A, to increase palatability, or to improve protein content in B. You can type B, or you can type C, cat. If you mention that primary goal of silage could be preserve forest under anaerobic condition, or the last one, silage process could be reduce fiber. So you can type A, B, C, or D. Let's see. But I like C. Yes, the dog from from Amanda Sophie the dog to reduce fiber. C, C. Yes, almost C. Ahmad C. So this the question we mentioned the main or the main objective to make the silage process. But others like A, B, D might be considered to secondary goal. Okay, so almost provides C, right? Primary goal of silage process is to preserve forage under an aerobic condition. Okay, C, right? Yes, everyone correct. So move to another question. So when we talk about silage fermentation or silage process, which group of microorganisms is the most beneficial for silage fermentation? So please type the list here. Yes, B boy, B boy. Lactic acid bacteria. So we can talk in short like lab. B, yes, good. Yes, almost we agree with lactic acid bacteria. Yeah, the answer is B, lactic acid bacteria or lab. But another microorganism might be contained in the silage fermentation process, but we did not uh, this microorganism occur in our silage. Okay, let's try the last one. The last one, when talk about silage quality, which fermentation products is an indicator of good lab activity in silage? Yes, D, dog. Any more? Yeah, D. Good. Oh, my friend from Uganda, that you choose the dog. Lactic acid is the fermentation products from activity of lab. Yes. Okay. Okay, thank you very much. So the answer is that lactic acid bacteria is the correct answer. Let's reply to the fermentation products. That is the indicator of the good quality of the silage. So thank you very much for your joining my question, and I think everyone can have good background already. So I would like to start with the importance of the silage. that that I will show you today. So this one is very important, essential for especially for ruminant production in tropical area. Particularly the silage can be served uh, for feed availability year round. We don't uh, much concern in raining season because green gas are available. However, in dry season, low quality lactate is the main feeding to the animal. So it might be not sufficient nutrient for the animal. So if we can making the silage in our country, so it may enhance livestock productivity since the animal consume the high quality lactate from the silage. So it can be uh, effect on the beneficial income in our farm. Several forest crop has been used in our country, such as corn. Corn is the ideal lavage source to produce the silage due to its high quality like plus protein and energy source, particularly water-soluble carbohydrate. In addition, another source like Napier gas, sorghum, or residue from cassava production, like leaf, can be also making in the silage to supply the lavage to the animal during dry season. Napier gas contain about 10% of crude protein and sorghum about 6 to 10%. In addition, cassava leaf, after we correct root to produce cassava chip or starch for the animal or human, leaf can be generated, and this one can uh, preserve like a silage diet and feeding to the animal. However, content of the cyanide may be effect to the animal. So before feeding to the animal, should consider the proper method to the animal. So let's now review the basic background of the silage production. So this section will cover the how fermentation work, which microbes are involved, as well as the condition, which condition microbes in the silage they need. Then we can manage those conditions in order to support the fermentation process complete. So this is some the uh, principle of silage. So this slide summary of four essential principles for successful silage production. First one, you can see that when talk about the silage, an aerobic condition is very important in order to provide microbe. Namely, lactic acid bacteria can work well on the generate lactic acid to decrease pH. So oxygen must be removed quickly after the process of silage. If not, so spoil or some microbes may be make the quality of silage low than we expect. The temperature in our country, temperature should be moderate warmth. Actually, we require about 25 to 37 to support lactic acid bacteria growth and synthesis their lactic acids. Suitable moisture, this one is the main the criteria to make the silage. In order to high or low moisture maybe inhibit growth of lactic acid bacteria. So the last one is the uh, energy source like a sugar source that contain in plant. This is the source of the food for lactic acid bacteria. Low sugar or low energy or water soluble carbohydrate. Lactic acid might be low production and this cannot suppress other microorganisms. So we try to support this one, like bacteria, to produce more lactic acids, and this can suppress some undeserved bacteria that we don't need. So this diagram shows the silage fermentation process. So first, at the above, you can see that temperature is the main one that can be changed after silage process. At the beginning, like 3 days after silage making, pH can increase, not pH, temperature can increase to almost 32 degrees Celsius, and then it can be stabilized after 21 fermentation of silage. Furthermore, pH is the main indicator of silage fermentation. At the area of silage production, pH is still high, actually about 6 or natural, like that 6. Then after the fermentation process of silage, so pH will be rapidly reduced until 4.2 or almost 4 after 21 fermentation process. The reduce of pH might be affected by the population of lactic acid bacteria that can be increased from the uh, Thursday of ensilage. And this one, you can see that in the pink one, at the earlier fermentation of silage, you can see that undesirable bacteria like anaerobic bacteria can be caused. This could be due to during the earlier fermentation, oxygen might be contained in the floret. So aerobic bacteria can consume oxygen for their growth. After oxygen removed or empty, so this bacteria, aerobic bacteria, can be reduced. In alternatively, lactic bacteria can be increased and improve the pH to be lower, as you can see in the right hand side. So sugar or water soluble carbohydrate reduce from the beginning. This could be due to the increasing of population of lactic acid bacteria that can synthesize lactic acids. This can be reduced pH. In short, we would like to get to quick pH reduction and lactobacillus bacteria synthesize more in order to preserve our silage and maintain the quality. So this slide shows the difference. There are many factors influence on silage metabolism during ensiling process, like silage additive or other approach can be support the quality of silage that uh, provides. First factor like the quality of forage crop. It should be contain more water soluble carbohydrate that can be supplied for lactic acid bacteria to their ghost or synthesis in their cell. Moisture should be suitable for the fermentation type of forage, buffer capacity. That is the factor can be affect to the silage quality. In addition, technology to making the silage like a packaging, searing of silo, or maintain air condition, that is the factor to improve the quality. In another way, we try to reduce storage loss. This can be retain the high crop feed value that will be feeding to the animal. So the last one, how to improve the quality of silage process. So it should try to create a lab population. In terms of high lab population, so it can be inhibit of Clostridia or fungi growth that can keep the quality of silage longer than we expect. So this table shows some the common fermentation and end product that we expect when compared with an uh, and low quality of forage like high moisture or so dry. This figure show the pH that you can see actually pH we need about we require about 3.7 to 4.2. In terms of the forage contain high moisture, it can be increased pH and higher than we expect. So this can be affect to the quality of silage. In addition, lactic acid we requires high content due to it can be control the pH as we need. In other criteria like a butyric acid, you can see that when suitable condition can be provides butyric acid. This is the product from undeserved microbe, so it should not contain in the silage. In terms of the high butyric acids might be reduce the quality of silage as well as ammonia, nitrogen. This indicator can be determine the quality in terms of quality, in terms of the crude protein contents might be break down into the ammonia. So it's actually ammonia nitrogen should be lower than 10, like mentioned in this table. So when we talk about the silage process, microorganism is the main actor can be produce the quality to be high as we expect. Several microorganisms that occur in the silage process like lactic acid bacteria, butyric acid bacteria. So those species they did not require oxygen. So the optimum temperature should be around 17 to 37, and optimum pH should be 3 to 8. However, the butyric acid bacteria like generate from bacteria group of Clostridia that we no need. So if we can lower this species of bacteria, so the quality of silage can be improved. And some bacteria or microorganisms like a sporate or Saccharomyces cerevisiae, mold and fungi. So those species they require oxygen. In case we not well prepared or packaging or selling, so those species can be occur and may spoil it of the silage. So this is still the challenges for animal production, particularly when we talk about making the silage in tropical area. There are several factors that we need to concern uh, like a temperature, high ambient temperature. So this factor may be the the quality lower than we expect. If the temperature higher than 40 degrees Celsius, after silo opening, it may reduce the quality or spoilage may be occur. And another one like infrastructure or technology, we did not have some big machine or sealing packaging machine. This can be provides oxygen into the silage and can generate the spoil in the products. And another one like quality of forage content like sugar or high fiber. This may be effect on the lactic acid production or pH did not reduce as we expect. And another one, high moisture content. This could be well considered. The higher than 80%, this may be lower quality of silage. And the seasonal availability, this may be considered as the quality of silage low. So based on the five criteria challenges, we have some proposed methodology to suggest in order to improve the silage quality in our country, like can be as some inoculant like bacteria, or can be as some feed additive in the silage products. So when we talk about bioactive compound, what is the meaning? Bioactive compound is the products that can be occur in the natural. So in some fruit, some vegetable, or earth, or grain that can be found. For example, like flavonoids, tannins, saponin, or essential oil. So these bioactive compounds can be effect on gut health, antimicrobial, anti-parasites. So this compound we expected that when addition to the silage, it may be break down or kill some undeserved microbe that we don't need, and it will allow lactic acid bacteria to generate more lactic acids and enhance the quality. So again, based on my previous presentation, which one is not the class of plant bioactive compound mentioned in the last presentation? You type A, flavonoid, cellulose, tannin, or saponin. Please type. Yes, yes, my friend from Uganda and Pakistan. Yes, B. Okay, in order to save the time. B, cellulose did not bioactive compound. Cellulose is the part of fiber segment. So let's see some the source of bioactive compound that we can find in the tropical area. I will start from flavonoid. So flavonoids can be occur in leaf, grain, and some peel. For example, moringa. Moringa is the tropical plant. So it's contained about flavonoid 2.6%. In addition, Leucaena, the one of a legume plant, it was found that in leaf contain flavonoid about 2.2%. Sorghum and green tea or citrus also contain flavonoid in the suitable component that can be effect to the microbe as we expect. So talk about tannin, where we can find the tannin in tropical plants. Firstly, cassava leaf contain condensinin about 1 to almost 4% dry matter. And also Leucaena again can find condensinin about 2%. Calendra and Sesbania in the quart meal and leaf, so can find quite high concentration of tannin. So those the plant source can be potential to extract to get some the content of condensinin in order to add to the diet or silage to improve the quality. Saponin as well can found in some like sesbania, alfalfa, or jam tuber, cedar. So the rank of saponin can be vary from 1 to 19%. So these several plants can be considered to get the phytonutrient compound to improve the quality of the silage. So what happened when we addition plant phytonutrient compound? So you can look at here both a dose of flavonoid, tannin, saponin, or some essential oil like from eucalyptus leaf or some garlic that can provide some essential oil, maybe effect on the silage quality, and finally they can improve the luminant performance. So when addition plant phytonutrient compound, we hypothesize that the quality of silage can be improved by reduced proteolysis. This can be act on lower ammonia nitrogen in the products. Furthermore, the addition of plant secondary compound can improve the lactose, prevents nutrient rot, and inhibit some undesired microbes like Clostridia, yeast, and mold. So another factor that we need the silage should be has more aerobic stability. So we expect that if plant phytonutrient compound addition, the silage can be kept longer than we need. When we check with the quality of the animal response, so ruminant can be get more undegradable protein. This might be based on the condensed tannin can be prevent protein digest. Fiber digestion can be improved. That this is the indirect that we need from the silage. High nutrient absorption efficiency and try some microbe that we don't need. Finally, some report may be report like addition of plant phytonutrient compound can be reduce methane emission in the ruminant. So when we look at the detail with how plant active compound affect on microorganism, it can be shown that this compound can affect on membrane disruption by interfere the pump of the ion like potassium, hydrogen, or magnesium can be linked via the membrane cell. In addition, plant compound can affect on inhibit cell growth or DNA synthesis. So this factor of phytonutrient compound can work and then finally can improve the quality. So another mode of action with addition plant phytonutrient compounds can be reduce methane emission, like you can see in this figure. After fermentation of wheat, so phytonutrient compound can inhibit methanogen bacteria, so methane can be reduced. There are several methodologies like plant can be effect on membrane cell of methanogen or suppress protozoa that they stay with the methanogenic bacteria. In addition, plant phytonutrient compound may support or suppress hydrogen producing bacteria, so it can be produce more propionate. This can reduce methane emission. So let's move on the effect of plant bioactive compound supplementation in silage. Then we check with the silage quality what happened for their quality. So you can see here, tannin 2%, condensed tannin 1.5 to 4%, inoculant into the silage like maize, alfalfa, moringa, or some legume. The results show that addition of tannin or plant secondary compound can be reduce pH. Reduce pH then is mean that the quality of silage may be improved and reduce ammonia nitrogen in the silage. So lactic acid bacteria almost they increase after addition plant bioactive compound. So some period experiments by my colleague, they try to make a silage by use clean manure hemp. So this plant contain contain about 0.1 to 0.4% of condensed tannin, which one contain about 12 to 15% of crude protein. When making into the silage form with the addition some enzyme or bacteria, the quality of silage can be enhanced, particularly lactic acid concentration, and can be low pH. And some sorghum that contain condentin about 0.3 to 1.5% when making into the silage. So the quality of silage can be improved by lowering pH below 4, high lactic acid content. And when checked with the quality with the digestibility, it was found that increased in vitro digestibility as well as methane reduced as well. So when look at the plant bioactive compound supplement into the silage material, then determine the rumen fermentation parameter. So several plant bioactive compound adding to the silage can be enhanced in vitro fiber digestibility as well as reduce ammonia and nitrogen. Furthermore, methane can be reduced by Shinas tannin or hydrolyzable tannin addition into the silage. This may be the effect of tannin inhibit the methanogenic bacteria. Then methane can be reduced. So this some my colleague demonstrate that Firmicea microphylla making into the silage. This plant contain quite high content of tannin. After ferment for 14 days or 2 weeks, it can be enhanced in vitro degradability as well as propionic acids. In addition, methane production of protozoa can be reduced when determined by using in vitro experiment. So this leaf generate from cassava that we know that contain quite high condentin of almost 4% and high crude protein. When fermented for 20 days, it can be enhanced crude protein content and increase dry matter digestibility. So table 7 show the effect of plant bioactive compound on digestibility and rumen fermentation. It was found that in beef, cattle, or in dairy cows fed with silage material, the results show that dry matter intake or ammonia and nitrogen can be enhanced, whereas methane production can be reduced when supplement with silage at the last source. When we look at about milk yield and milk composition of dairy cow when feeding with the silage inoculant with plant bioactive compounds, in dairy cows fed with lucerne can be enhanced crude protein content, fat, and lactose. However, milk yield did not change both in dairy cow and goat fed with lucerne silage or cassava. Furthermore, in Holstein Friesian feeding with the alfalfa fermented or making the silage with tannin at 1.6%, the result demonstrates that milk yield and milk composition, protein, and lactose has been increased when compared with the normal silage without tannin supplementation. So these results demonstrate that the useful of silage fermented with the plant compound may be effect in the positive on milk yield and milk composition. So some byproducts like durian peel can be made into the silage since it contains like 4 to 5% of tannin. After silage, it can be enhance the quality of silage, and goat can be good response on increased digestibility, better C3 or propionic acid, and reduce methane production. In our country, we demonstrate that the byproduct from palm production, like palm frond silage, this contain about 6% of condentin. After making into the silage, it can be enhance the quality silage and enhance the utilization of feed in goat and enhance the fermentation. And the earlier publication in Animal Bioscience by our colleague, they try to making a silage by use leaf and root combination each other. So those products contain tannin about 1 to 4%. So this they making the silage by use root 40% and leaf 60%, ferment for 14 days, and the results demonstrate that the quality quite good when checked with the silage quality and response of beef. They demonstrate that this product can be replaced concentrate up to 50% without adversely effect on beef performance. So this is my last presentation slide. Addition of the bioactive compound is the alternative method to improve the quality of silage. That can be enhance the quality, improve the condition, as well as the silage can be longer preservation. When look at the plant bioactive compound supplement, it can be referred to reducing proteolysis that can provide more protein or amino that supply for the animal. It can reduce nutrient loss. When feeding to the animal, beef, dairy cows, or goats, it can enhance rumen fermentation, nutrient absorption. Finally, the animal products like milk yield and milk composition can be increased when feeding silage with plant bioactive compound addition. So if you would like to read some our research or work, you can go into Scope like a Scopus database or ResearchGate. Here you can scan QR code and reading more the paper that we have done, not only silage making, but also in other key elements or other knowledge like methane emission or utilization of byproduct into the animal fish. So thank you very much for the organizing committee IPB University to present to invite me to present the knowledge, and thank you everyone for your kind attention.
Thank you, Professor. It was a very interesting presentation. Professor Ansukman include a quiz that actively engage the participant. Thank you, Professor. This question from Balalai Fall. At what stage of silage making do I add the PBCs? Please directly to answer, Professor.
So the question at the what stage? So we can add directly at the beginning when we process the silage. You can chop the forage and check with the quality of moisture, content of moisture. Then during you start to do the silage, you can add the plant bioactive compound. So another question, right? From Salifa, how can systematic identify?
From
Salifa. How can we systematically identify catalytic effect of plant compound? So you mean that how can we identify the mode of action of plant, right? How can we inhibit growth sporate, right?
Sarifa, can you directly ask to Professor?
Yes, sir.
Please, Sarifa.
Okay, sir. How to systematically identify and catalyze the most effective plant bioactive compounds to inhibit the growth of spoilage microbes in process spoilage, in tropical spoilage.
Mhm. So we can analyze, we can check with some like a machine to analyze the content of bioactive compound, right? For example, the machine like you check the content of compound in plant like high performance liquid chromatography that can be determine the content of bioactive compound, and then you can after that you can check with the quality of silage. You can check with the population of microbe, how many they can grow, particularly lactic acid bacteria they grow in our product, whereas some Clostridia or some mold or yeast we don't need, so we can count on their cells by use like uh, real-time PCR, or you can culture by page. Some plant compound you can check directly. So firstly, you can grow bacteria like Clostridium or Bacillus or more. Then you can add this bioactive compound in the solution. Then if this bioactive compound can prevent their growth, it means that your bioactive compound can act very well on prevent their growth in your silage. Okay.
Okay, thank you, sir.
Okay, Sadipa. Thank you.
Okay, move to another question from Akira, right? From Sir
Aidan,
how can metagenomic approach to apply you evaluate the plant signal? Yes, good. So this quite modern technology to apply metagenomics to evaluate the effect of plant secondary compound. So this method can be allow us to check with the number, right? Particularly the DNA of bacteria that can grow to the silage. So you can collect the sample of the silage and then analyze by use this method and check with what kind of bacteria or microorganism you need. You can directly count by this methodology. And the second question, the application of essential oil for silage preservation. Yes, the mode of action of the essential oil quite similar with other plant secondary compound. So it can be effect on the cell membrane or inhibit the DNA synthesis or cell growth. Another technology like a nano encapsulation is quite good due to some we concern that when feeding 100 oil, this may be effect to not only undesirable microbe, but it may be effect on lab bacteria. So nano encapsulation may be alternative approach to prevention the effect of oil on both lab and undesirable microbe. However, when talk about the micro encapsulation, the price might be considered. Okay, another question.
There's question from Sahil Jahman.
Saman, right? Saman. So when adding the condensated or plant secondary compound, how do rumen microbiota adapt over time? So when talk about the inoccurrence or addition some feed outside into the rumen, the rumen microbe they quite well adapt by themselves when we addition of tannin or saponin. It did not add only one time that's sufficient, but we need to add several times. For example, 1 week a time or 2 weeks in order to allow plant secondary compound act as we need. So if we cannot add only one time, that sufficient. These prebiotics that can be.
Okay, Liana Wahyuni, please.
Thank you, sir. Good afternoon. My name is Liana Wahyuni. I as a feed quality supervisor at the Agriculture and Livestock Department of Musirawas Regency Government. So I'm so interested with your topic. So, sir, every plant contains secondary metabolites, including tannin, saponin, flavonoid, and the others. As far as I know, that compounds have several benefits for livestock, such as reducing methane emission, antioxidant, and so on. So, however, these compounds can also be anti-nutrient. So, my question is, what percentage of these compounds to be anti-nutrients? Thank you.
Yes, that is the quite good question. So actually some plant is contain quite high composition of anti-nutritive substance like tannin and saponin. So they have the recommendation by previous researcher contain tannin or anti-nutritive substance should not over than 4% of the diet that animal consume. If the high concentration higher than 4 or 5%, the animal can less consume of feed, right? And can be effect on microorganism, both beneficial species. So finally, the animal can generate low productivity. Mhm. Based on my presentation, when talk about the silage making, like a tannin, saponin should not higher than 4 or 5%. Mhm. So before correct the forage and then before making the silage, so we need to check how many percent of tannin of the in contained in our forage before making to the silage.
Okay, for the last question, there is also from Balalai Fowl. The last question. Are there prebiotic that can be added to silage to support LAB and other bacteria?
Good question. So yes, it can be added in order to support the lactic acid bacteria. For example, like some plant bio, some forage, it may contain the low low carbohydrate or low vitamin or low some prebiotic we can add in order to support the desirable bacteria that we need. We can add.
Okay, I think time is out. Thank you very much, Professor Anuson, for your insightful presentation and the engaging discussion. We truly appreciate your contribution. Thank you, Anuson.
Yeah, thank you very much.
Okay, we will now immediately continue with the second speaker. So we kindly ask all participants to stay with us and remain in the session. There is more valuable knowledge coming your way, so please don't go anywhere.
Asep, who will share the PowerPoint? Excuse me, Pak Asep. Pak Asep.
Okay, Pak Asep Sudarman.
Okay, thank you very much.
Udah oke ya? Before we begin, let us now begin with our second speaker, Professor Dr. Insinyur Asep Sudarman, Master of Rural Science, a professor at the Faculty of Animal Science, IPB University Indonesia. Professor Asep completed his bachelor degree at IPB, a master in sheep nutrition from the University of New England, Australia, and a PhD from Hiroshima University, Japan. He specialized in rumen fermentation, sustainable feeding system, and ruminant physiology. Today, he will present foster the development of Garut sheep for the livestock industry in Indonesia. Please welcome Professor Asep Sudarman. Professor Asep, the screen is yours.
Excuse me, Pak Asep, who will share the PowerPoint? Pak Asep.
Okay.
Who will share the PowerPoint? It's me.
Prepare. Okay, Professor Asep, please can you share it by yourself?
Okay. Can you see the PowerPoint?
Yes. Yes, it's visible.
Okay, I have to full screen first. Where is it? Where is the instruction? Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and all participants for various countries. It's a pleasure to meet you all, and I hope everyone is doing well today. I've been assigned to deliver a presentation entitled Fostering. What is that? Fostering the development of Garut sheep for the livestock industry in Indonesia. On this occasion, actually, I will not only discuss Garut sheep for meat production, but also for sheep fighting competition, which are part of the culture of the West Java people. Garut sheep is Indonesian exotic sheep. It is established in 1864 in Garut Regency of West Java Province. And Garut sheep are the result of crossbreeding between 3 breeds of sheep. Those are Merino, and then Cape Fat Tail from South Africa, and Japanese Tin Tail sheep. The characteristic of male Garut sheep, as you can see in the picture, they are characterized by its large body weight, with adult rams typically weighing between 60 and 80 kilograms. Its most distinctive feature include large spirally shaped horn and strong neck. The ears of Garut sheep are notably very short, and its tail is short and enlarged at the base. The sheep come in various colors, including white, black, brown, or combination of these three colors. As I said before in earlier that Garut sheep also used for fighting contests, and the champion from this contest usually has extremely high price, often exceeding 100 million rupiah, or it is equal to maybe $6,800. While adult female sheep typically weighing between 30 to 40 kilograms, unlike some other breeds, they don't possess horns. While the sheep are prolific breeders, but farmers often prefer ewes that give birth to only one lamb, as the limited milk production of the ewe means only a single lamb is likely to survive. Next is performance of Garut sheep. This table shows the birth weight with different number and sex of lamb per bird. The highest birth weight is in single bird type, followed by twin, then triplet. And male usually has higher birth weight than that of female. This is single, twin, and triplet. You can see here, the single is bigger than twin and triplet. During the growth period, the highest daily gain is reached at age 30 months and 3 weeks old. And at this time, feed efficiency utilization is also reached the peak. After that, the growth rate declined. The farmer raised Garut sheep is first for meat production purpose, and the second is for fighting contests, as I mentioned before. For meat purpose, there is no special treatment given to sheep, and the price of sheep is normal according to kilogram body weight. And for ram fighting sheep, there is special treatment given to sheep to become champions, and the price of sheep for this purpose is very expensive. There are two farming systems or objective of rearing sheep run by the farmer. That is for saving as a traditional system and for making profit business or making profit or business. Rearing sheep for saving is characterized by low input production, low technology application, and selling sheep when farmer needs money independent on the price. Rearing sheep for making profit is characterized by use business input rationally, implement technology even if it's simple, and selling the sheep regularly. Feeding system is applied by the traditional farmer usually based on the tradition and custom of their parents, not according to sheep nutrient requirement. And types of feed given to Garut sheep are based on the availability in local area, such as field grass, banana leaves, jackfruit leaves, commercial concentrate, rice bran, tofu and tempeh byproduct, coconut milk byproduct, and cassava, and etc. The farmer usually fed their sheep 3 times a day. In the morning, they give grass, usually field grass, and 2 hours later, the sheep are given concentrate feed, such as tofu waste. And in the afternoon, the sheep fed grass ad libitum. Garut sheep, especially male sheep, prepare for fighting contests, are given special feed and meet good nutritional value so that they have a good appearance. Feed are given to them 2 times a day. Amount of feed given is adjusted to the weight of the ship so that the ship are not too much fat and therefore the ship can move freely during the competition. Ship prepared for fighting competition are given supplementary feed, particularly with an energy source that is cassava tuber. Sheep are also given special supplement, usually eggs mixed with honey. Supplementation can be given 2 days before competition. Then the sheep are rested 1 day before the competition. And traditional medicine, or we call usually jamu, a medicinal herb, are also given. Fighting sheep are given several training sessions aimed to accustoming the sheep's muscle to strength and having optimal power when competing. Running is done every day for 15 minutes, and swimming is done once a week. As you can see in the picture here, this is running done every day for 15 minutes, and swimming is done once a week only. And training to fight is done only when the ram has reached 3 years old. The first fighting exercise is done no more than 10 collisions, then increase gradually until it matches the class of sheep. There are 3 classes of sheep. Class A weighing 60 to 80 kilograms, Class B weighing 40 to 59 kilograms, Class C weighing 25 until 39 kilograms. For B and C class, they are trained as many as 20 collisions. And for A class ram is up to 25 collisions. Garut sheep, especially for fighting, receive regular care from the farmers. Shearing wool every 6 months, and some are less than that. Cutting the hoof or toenail every 6 months, every 4 months. Garut sheep has 4. What is this? Sheep also being bathed once a week on Friday. Bathing the sheep take about 15 minutes using liquid soap through rubbing the entire body. Sheep also usually gets massage therapy before bathing. And this is the type or the shape of Garut sheep horn. Yeah, there are 4 shape type of Garut sheep horn. On the left top we call gayor, and on the right top we call golong tambang, and for bottom left we call leang, and for bottom right is ngabendo. All horns look beautifully symmetrical. However, not all of these horn shapes occur naturally. Asymmetrical horns are corrected by a horn repair specialist. Asymmetrical or irregular horns need to be repaired because it is affected health, selling the price of sheep, and for ram contest will reduce the assessment. The age of sheep to be repaired their horn no less than 2 years old. You can see the picture here. This is before repairing the horn, and this is after repairing the horn. Yeah, the distance, this is narrow and this is far enough, become far. And there are 4 steps in the process of repairing asymmetrical horns. First is preparation. The sheep is secure with protecting its face with a damp cloth, and a banana stem is used to prevent horn pressure. And then the second, softening. Horns are coated with soap and soy sauce. Then they heat it with fire for 10 to 15 minutes to soften. And the third, shaping horn. Iron clamps is used to slowly straighten or shape the horns. And the last is finishing. The process is repeated until horns are symmetrical. Each correction takes about 30 minutes to 2.5 hours. And these are photos from a sheep fighting competition. The top 3 photos show sheep preparing to fight. And the bottom 2 photos show the sheep were fighting. The level of this competition usually varies, and the top one is Presidential Cup. Now we come to Garut Sheep Breeding Center. It is located in district of Margawati in Garut Regency. It is about 250 kilometers from Bogor, where IPB University is located. The centers currently manage about 1,500 Garut sheep, which are capable of producing around 600 superior quality breeding stock annually. And sheep produced by the center are distributed to farmer groups from underprivileged communities, and the second to independent farmers spread across various regions in West Java. The number of sex ratio of sheep distributed are 1 ram to 10 ewes per farmer group. For the sake of Garut lamb meat production, it needs to be upgraded. This is the existing condition of Garut sheep. First, the Garut sheep exhibit lower in growth rate, reproductive efficiency, also lower in reproductive efficiency, and carcass yield when compared to specialized commercial breeds. And the second, there is no organized breeding programs. Therefore, it needs strategic crossbreeding and genetic improvement that is crossing Garut and Dorper sheep. And this is the result of crossing Dorper and Garut sheep. The crossbreed looks has more body conformation and better amount of flesh compared to origin Garut sheep. And we can see the production performance of crossbred Dorper and Garut. Birth weight in all criteria is higher in crossbred than those in Garut sheep. Winning weight is also higher in crossbred than those in Garut sheep. We can see here, for example, male in crossbred is 16 kilogram, and here is about 14 kilogram. Female is 15, and here is 14. And then this is the birth type, single, multiple, and multiple. Also, Garut sheep is lower than crossbred. And pre-weaning growth is also higher in crossbred than those in Garut sheep. Here, male has 145 gain, and here only 108 gram. This table shows that except for litter size and reproductive index, other parameter performance are better for crossbred than Garut sheep. You can see here, for lambing interval day, Dorper sheep crossbred is 214 days, and Garut origin 211. And litter size, Garut sheep is better than crossbred, and mortality, crossbred is better than Garut sheep. And reproduction index, Garut sheep is better than crossbred. And productivity index, crossbred is better than Garut sheep. And this one of the biggest commercial sheep farming. PT Agro Investama is located in Malangbong district of Garut Regency. It is about 200 kilometers from Bogor. They have a farm area of 60,000 meters square, and they possess sheep in 2018 about 1,900 heads, and 2020 is 4,200. And also they have full blood dorper, about 100 heads. These are imported from Australia. You can see the picture here. These are full blood dorper, and this is the crossbred one. Yeah, this is also the crossbred. PT Agro Investama operate its business with modern management. Sheep are fed total mixed ration, which is mixed and distributed using a special mobile feeder. We can see here this. They mix using the special tools, and then the total mixed ration distribute using also this one. Okay, and the photo on the bottom right is a lamb chop from a crossbred dorper and Garut, which has a very delicious taste. Yeah. Okay, I think that's all. Oh, sorry. That's all for today. Thank you for your attention. Thank you very much. Pak Asep, please.
Yeah, thank you, Professor Asep, for your insightful and inspiring presentation on the development of Garut sheep. Your work clearly highlights the unique role of local livestock breed in both cultural preservation and modern industry. We now open the floor for questions. Participants may use the chat box or raise your hand to speak directly. Okay, Mr. Jaidan, please.
Hello, Professor. So thank you very much for your insightful presentation. I found that traditional feeding practices and physical training programs for Garut fighting rams very fascinating. So I would like to ask, like, considering the spatial feeding regimens, including industrial byproducts and herbal supplements like jamu, do you think there's potential to apply such like a study in nutrigenomics to evaluate how such diets affect the gene expression related to muscle development, energy metabolism, or stress adaptation in fighting rams? And as follow-up, could gene expression profiling or biomarker discovery be used to support selective breeding of rams with superior contest performance traits? I'm very interested in how traditional knowledge could be integrated with molecular breeding tools to improve garrotship in a targeted data driven way. So I already list my question in the comment section below. Thank you, Professor.
Can I answer directly, Pak Asep?
Okay, please, Professor.
Okay. So far, what I know, the topic you asked has not been done in the research, and therefore this topic is very potential to be done. Maybe you can, if you make a proposal and you can propose to many what the fund source, yeah, fund source, and maybe I hope you can get the money for doing that research.
Okay, thank you, Professor.
Okay, enough, Ms. Kaida?
Yeah, I think that's enough.
Okay, I invite for the other question from the other person, all participant.
Zaidan, you actually where what university you come from?
Actually, I'm studying in biotechnology information at National Taiwan University. So right now I'm not in Indonesia.
Oh, okay. So you can go back to Indonesia and doing the research about genomic for that for
Yeah,
fighting REMSIP.
yeah, I think so. I will back to Indonesia for the summer IPP University excursion on 20 July. Maybe I will go to IPP and see what happening there.
Okay.
Thank you, Professor.
Okay, I invite for the other question, please. There is a question from Fadila. Please, is there any case of reported injury or damage to brain or health in this animal? Fadila, can you directly ask Professor Asen?
Who is he? Where is he? Where is he? Can I open? What is the question is? Where is it?
Fadila, can you hear me? Please open your mic before you say.
My question is like, is there any reported case of brain damage or health issues since the animals are kept for the show? And what is the maximum age for which you can keep these animals for replacement?
I cannot catch your question.
Like, what is the maximum age that you can keep the animals before you can replace them, or that since the animals are kept for show, is there any health issues? What are the health issues?
Sep, can you explain?
Like, since they are used for fighting and they use the head to fight, so I was thinking with time, maybe that activity can cause maybe damage to the brain or any health
Sep, can you
issue.
explain? Can you repeat the question?
I said, is there any
So, Mike, you still unmute the mic. Sep, your mic still
Okay.
unmute.
Please, is there any case or reported injuries or damage to brain or health in this animal and what is the maximum age that this animal can be kept for the soul?
Okay. In short, maybe it is dangerous or not for the ram. That's right. Actually, in nature, the ram also has a habitual to fighting each other, and I heard that the structure of the skull of the ram is different with other animals, and the other is that in this tradition or culture, usually this is not freestyle until the animal collapse. The collision is only the maximum collision is only 25, and usually if there is one of ram collapse, usually those maybe we call the winner in in quotation mark will be will be eliminated. So the committee maybe usually they try to pair two rams to fight with the same level. Yeah. So I haven't heard about the what you call the ram become dead due to the fighting in this contest. Okay, I think that's the answer.
Yes, thank you.
Okay, I invite for the other question, please. Okay, while waiting for the question, allow me to ask one or two the discussion. Professor Asep, you mentioned that Garut Sip had a strong cultural role in West Java through ram fighting contests. In your view, how can this tradition be maintained while also ensuring compliance with animal welfare standards that are increasingly emphasized globally?
Yeah, I think the question is almost similar with previous question. Actually, there is regulation in what regulation created by the association that they try to avoid the animal become collapse. Yeah, and therefore the coalition is limited as much as 25 coalitions. That's for class A. Yeah, for class A. But for lower class coalition is maybe also lower than 25 coalitions. And as I said before, that the skull of the head structure of the sheep is different with other animal. Maybe you have ever seen the sheep collision with the bull, and the bull become collapse. In the YouTube, you can see in the YouTube.
Okay, because now there is animal welfare is very interesting. So maybe for what about or involved in animal welfare is very a little bit cruel for the fight this fighting. I have a second question. You briefly mentioned the crossbreeding effort between Garut and Doper sheep. Could you share more about the observed performance improvement, especially in terms of growth rate and carcass quality?
Yes, that's true. The crossbred between Dorper and Garut sheep produce more carcass and not just quantity but also quality. And you can try the lamb chop in steakhouse in Bandung. I have tasted for several times. And I love that lamb chop.
Okay, thank you,
Pak Asep,
Professor. I invite for the other question
Yeah,
from the
there is one
audience.
question. I think there is one question
You can
from
raise your hand or
Stephen Zidane.
chat in the chat box.
Is there any question?
Okay, if no respond, I think it's enough for this session. Thank you, Professor Asef, for the nice presentation. Thank you for all.
Thank you very much.
I will return to MC Dara, please.
All right, thank you so much, Dr. Asep, for moderating today's session very smoothly. Also, thank you for Professor Asep Sudarman and Professor Anusorn for your engaging and insightful presentation. This concludes our day 4 of our summer course. I want to reminder For our participants, don't forget to fill out the attendance form and complete the tasks related to today's topic, which is available through the OCW. Our next course will be held tomorrow at 1 p.m. So I think that's all for today. See you tomorrow. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.